Hello students, I am Dr. Tisha Vyas. Today we are going to have a look at the second part of the history of biotechnology. So let us have a look at the different stages that took place in the development of biotechnology. There are three stages. The first one, that is ancient biotechnology, is the early history that is related to food and shelter, which also includes domestication. The second phase is the classical biotechnology that has been built on the ancient biotechnology, which also promotes processes like fermentation, food production, and medicine. The third phase is modern biotechnology. It basically means manipulations that has been done in the genetic information in the organism that you can also call as genetic engineering. Now, let us have a look at the difference between the traditional biotechnology and the modern biotechnology. Traditional biotechnology. It refers to the traditional techniques of using living organisms to yield new products or modify foods or other useful products for human use. Whereas modern biotechnology is the diversity of microorganisms and development of genetics expanded the potential of traditional biotech and ultimately that lead to the development of modern biotechnology. The examples of traditional biotechnology it includes recombinant DNA techniques, tissue culture, and mutagenesis, whereas modern biotechnology includes DNA profiling, genome analysis, transgenesis, DNA cloning, tissue engineering, etc. Traditional biotechnology includes breeding animals and crops to make cheese, yogurt, bread, beer, and wine. Microorganisms are used to produce various products such as enzymes for use in laundry detergents. Whereas modern biotechnology are used in many areas such as food, agriculture, forestry, healthcare, environment, minerals, and in industrial processes. Traditional biotechnology involves use of natural organisms to create or modify food or other useful products for human use, whereas modern biotechnology involves manipulation of genes and living tissues in a controlled environment. Now, let us have a look at the different areas of biotechnology. Different areas of biotechnology basically means that different kinds of organisms are used in making manipulations. The first one is orgasmic biotechnology, which basically means that use of the intact organism as it is, and it does not involve the alteration of its genetic material. Whereas molecular biotechnology, it alters the genetic makeup to achieve specific goals. There is another category that is transgenic organism, which basically means that an organism with artificially altered genetic material. There are several benefits of biotechnology, like in the field of medicine, which includes human, veterinary, biofarming, in the field of environment, agriculture, food products, industry, and manufacturing. In the next coming slides, we will be having a look at the different scientists that contribute majorly in the development of biotechnology as a subject. The first one, which you can see over here, is Anton van Leeuwenhoek. He was a Dutch businessman and scientist in the golden age of Dutch science and technology. A largely self-taught man in science, he is commonly known as the father of microbiology and one of the first microscopists and microbiologists. Van Leeuwenhoek is best known for his pioneer work in microscopy and for his contribution towards the establishment of microbiology as a scientific discipline. He used single-lens microscopes of his own design, was able to experiment with microbes, which he also referred as diarchens, which means small animals. Through his experiments, he was the first to relatively determine their size. Most of the animalcules are now referred to as unicellular organisms, although he observed multicellular organisms in pond water. He was also the first to document microscopic observations of muscle fibers, bacteria, spermatozoa, red blood cells, crystals in gouty toffee, and blood flow in capillaries. The next scientist is Gregor Johann Mendel. Mendel was born in a German-speaking family in the Silesian part of the Austrian Empire, today known as the Czech Republic. He gained posthumous recognition as the founder of modern science of genetics. Though farmers had known for millennia that crossbreeding of animals and plants could favor certain desirable traits, Mendel's pea plant experiments conducted between the year 1856 and 1863 established many of the rules of heredity now referred to as the laws of Mendelian inheritance. Walter Sutton was an American geneticist and physician whose most significant contribution to present-day biology was his theory that the Mendelian laws of inheritance could be applied to chromosomes at the cellular level of living organisms. This is now known as the Boveri-Sutton chromosome theory. The next scientist, Thomas Hunt Morgan, was an American evolutionary biologist, geneticist, embryologist, and science author who won the Nobel Prize in Physiology or Medicine in 1933 for discoveries elucidating the role that chromosomes play in heredity. Morgan received his PhD from Johns Hopkins University in zoology in 1890 and researched embryology during his tenure at Bryn-Mawr. Following the rediscovery of Mendelian inheritance in 1900, Morgan began to study the genetic characteristics of the fruit fly Drosophila. In his famous fly room at Columbia University, Morgan demonstrated that genes are carried on chromosomes and are mechanical basis of heredity. These discoveries formed the basis of modern science of genetics. During his distinguished career, Morgan wrote 22 books and 370 scientific papers. As a result of his work, Drosophila became a major model organism in contemporary genetics. Scientist Ernest Ruska was a German physicist who won the Nobel Prize in Physics in 1986 for his work in electron optics, including the design of the first electron microscope. Ernest Ruska was born in Heidelberg, Germany. He was educated at the Technical University of Munich from 1925 to 1927 and then entered the Technical University of Berlin, where he posited that microscopes using electrons with wavelengths 1,000 times shorter than those of light could provide a more detailed picture of an object than a microscope using light, in which magnification is limited by the size of the wavelengths. In 1931, he demonstrated that a magnetic coil could act as an electron lens, and used several coils in a series to build the first electron microscope in the year 1933. Sir Alexander Fleming was a Scottish physician and microbiologist. His best-known discoveries are the enzyme lysozyme in the year 1923 and the world's first broadly effective antibiotic substance, benzyl penicillin, that is also known as penicillin G, from the mold Penicillium notatum in 1928, for which he shared the Nobel Prize in Physiology or Medicine in the year 1945 with Howard Florey and Ernest Boris Chain. He wrote many articles on bacteriology, immunology, and chemotherapy. Fleming was knighted for his scientific achievements in 1944. In 1999, he was named in Times Magazine's list of the 100 most important people of the 20th century. In 2002, he was chosen in the BBC's television poll for determining the 100 greatest Britons, and in 2009, he was also voted third great Scot in an opinion poll conducted by STV behind only Robert Burns and William Wallace. Scientist Rosalind Franklin was an English chemist and X-ray crystallographer whose work was central to the understanding of molecular structures of DNA, RNA, viruses, coal, and graphite. Although her work on coal and viruses were appreciated in her lifetime, her contributions to the discovery of the structure of DNA were largely recognized everywhere. James Watson, together with Francis Crick, were jointly awarded the 1962 Nobel Prize in Physiology or Medicine for their discoveries concerning the molecular structure of nucleic acid and its significance for information transfer in living material. The results were based partly on the fundamental studies done by Rosalind Franklin, Raymond Gosling, and Wilkins. Scientist Mary Claire King is the American Cancer Society professor of the Department of Genome Sciences and of Medical Genetics in the Department of Medicine at the University of Washington. She studies human genetics and is particularly interested in genetic heterogeneity and complex traits. She studies the interaction of genetics and environmental influences and their effects on human conditions such as breast and ovarian cancer, inherited deafness, schizophrenia, HIV, systemic lupus erythematosus, and rheumatoid arthritis. Sir Ian Wilmot is an English biologist, embryologist, and the chair of the Scottish Centre for Regenerative Medicine at the University of Edinburgh. He is best known as the leader of the research group that in 1996 first cloned a mammal from an adult somatic cell, a Finnish Dorset lamb named Dolly. Let us have a look now at the early biotechnology. In the early part of the 20th century, technoscientists began to see zymotechnology as included in the applied sciences analogously to chemistry. They established institutions for collecting microorganisms. The concept of zymotechnology was broadened to a general concept of biological chemistry, involving the use of biological molecules such as amino acids, proteins, and enzymes in the industrial production. The word biotechnology was coined by Karl Erecki in Hungary in 1919 to describe general processes of converting the raw materials into useful products, such as on the industrial farms. In Britain, Chaim Weizmann developed bacterial fermentation processes for producing organic chemicals such as acetone and called it propellants. During the World War II, he worked on synthetic rubber and high-octane gas. World War II is also known as the era of penicillin production. In 1928, Alexander Fleming accidentally discovered the properties of penicillium fungus but determined that it would not be possible to use it in medicine. In 1940, a team of researchers at Oxford University found a way to purify the penicillin and keep it stable. Pfizer, which has made fortunes using fermenting processes to produce citric acid in the 1920s, turned its attention to penicillin. The company developed a complicated production processes and opened a new factory to implement it. Pure strains of the mold were first grown in small bottles, then seed tanks, and then deep fermentation tanks. The mold was then purified using a crystallization process. Using this new method, they were able to produce as much in a day as they had in the previous year. This massive production of penicillin was another major factor in the Allied victory in the World War II. Over here, you can come across a picture showing a scientist working on the production of penicillin from Pfizer in the year 1944. Now let us have a look at the various visions of the scientists that actually came to come across recognition of biotechnology as a separate field. During the interwar period, philosophers, sociologists, and public intellectuals began to reflect on the growing link between biology and technology. They put forward the idea that biotechnology could be used to change human nature, and by changing human nature to change the society. The Austrian Real France, for example, claimed that we would regard life as a series of technical problems for which living organisms acted as optimal solutions. In Britain, biotechnology was conceived of as a possible solution to the damages of the Industrial Revolution. Patrick Geddes, the Scottish biologist, divided the history of technology into three stages. The first one, that is Paleotechnique, that is Industrial Revolution. The second phase is Neotechnique, that is again the Industrial Revolution. And the third one, that is the Future Industrial Revolution, also known as the Biotechnique. Now let us have a look at the Patrick Geddes construction of biotechniques. He explained that the environment acts through function upon the organisms, and conversely the organisms act through function upon the environment. In human terms, this can be understood as a place acting through climatic and geographic processes upon people and thus shaping them. At the same time, people act through economic processes such as farming and construction on a place and thus shape it. Thus, both place and folks are linked through work and are in constant transition. Patrick Geddes was influenced by social theorists such as Herbert Spencer and French theorist Frederick Lepley, and expanded upon earlier theoretical developments that lead to the concept of regional planning. He adopted Spencer's theory that the concept of biological evolution could be applied to explain the evolution of the society, and drew on lay-place analysis of the key units of society as constituting but changing the last from family to folk. In this theory, the family is viewed as the central biological unit of the human society from which all else develops. According to Geddes, it is from stable, healthy homes providing the necessary conditions for mental and moral development that comes beautiful and healthy children who are able to fully participate in the lives. According to the US concept of biotechnology, various fields are ultimately linked to the biotechnology field, right from the mathematics, applied sciences, experimental designs, decision theories, learning theories, aesthetics, vibrations, neurophysics, biophysics, anthropology, meteorology, human engineering, pharmacology, biochemistry, toxicology, biology, physiology. All the fields are interlinked to one another and ultimately they are linked to each other. Another scientist, that is Craig Taylor, is also a notable scientist in the field of biotechnology. Dr. Taylor and his research team were conducting a number of studies related to prevention and treating eating disorders. They took number of projects, evaluated and disseminated accessible evidence-based mental health care to large defined populations. They did number of e-courage projects and also had many collaborations with the healthcare department. Thank you all my students. This was all about today's lecture on the history of biotechnology. If you have any queries, you can mail me at my email ID which you can see over here, and you can also text me on WhatsApp. Thank you.